coming. I want to thank the ISB organizers and in particular the reviewers for the papers for selecting our work for presentation here. The title is up at the top, and this work was published earlier this year in PLOS Computational Biology, and the site is there at the bottom. As I was traveling to the conference this week, I happened to pick up a copy of the news magazine, and on the cover story said, "The genome, big advances and many questions." And I started reading with great interest. And about halfway through, we were reminded why studying in a translational science setting, why studying so many diseases is difficult, and that is because they are polygenic. But most interesting is right after that quote in the paper, or that paragraph in the paper, there was a quote by Dr. David Goldstein at Duke, and he said, with respect to the difficulty of finding candidate disease genes, he said it's like when a suspect is in the neighborhood but not knowing what house the culprit is in. And I thought that suitably rephrased as saying that we have no chance of finding what house the culprit is in if we're not in the right neighborhood. So that's what I'm going to show you today. It's a nice segue. I'm going to share with you today how a proteomics-driven approach can find candidate subnetworks in disease and find out what culprits are involved in disease. So by way of overview, I'm briefly going to share with you some background approaches for protein-protein interaction network-based models. I'll share with you then our proteomic approach with a brief discussion of our computational model, some results from implementing that model on data obtained in our lab and one other, some wet bench validation, comparison to a genomic-driven approach, that is using genes to drive our model instead of protein disease targets. I'll give you a dry bench validation, that is classification performance, and if time permits, a little bit on our latest work. So this is by no means an exhaustive, but I want to share with you some of the methods that have been published in protein-protein interaction approaches to finding candidate disease genes. The first one was done by Trey Eidecker's lab back in 2007, and he worked on a model of human and what they did is they obtained microarrays, transfer files from the public sector that had been detailed on experiments performed in human breast tissue between tumors and matched controls. They took those expression values and they painted them a human protein interaction network, and then by a greedy search, the entire global interactome, looking for small subnetworks that differentiate between tumor and control. Having that differential activity in those small subnetworks, they then use them as features classifier, which in a cross-validation setting was a more accurate for breast cancer metastasis than traditional approaches. Similarly, Vanunu and Sharan used disease genes, that is genes known to have a role in a variety of diseases. They combined these with PPI interactome confidence scores, and once again, a global search of a human protein interaction network, and produced a recorded list of causal disease genes. Many genes which came out that showed high rank of the genes that were in their seed, but also they were able to infer new genes and also infer new complexes in the disease I listed here. By a similar approach in 2007 showed that you could take genes discovered by linkage analysis, the seed random walk, go through an entire global PPI, and again use a prioritized gene list, which were quite few for positional cloning projects. Okay, so things to point out about these studies is that while they show varying finding candidate disease genes, all proceed with a genomic-based approach. In the case of Eidecker, he used profiling, and in the case of the other two, they used causal disease genes or genes found by GWAS study, mutated genes or genes found by GWAS study. Okay, so we got to wondering, well, what would happen if we seeded an approach like this with proteomic disease targets as opposed to genomically derived disease targets? So this is what we did. And our model is basically described here. We study 4 samples taken from colorectal biopsies, late-stage tumor biopsies with matched controls in patients diagnosed with colorectal cancer. We separated those by 2D page for spots which were up greater than 2-fold and were significant on the gel. We excised those from the gels robotically, sequenced them by LCMS, and ended up with 72 targets highly significant for the colorectal. Okay, we took those disease targets and used them to see a human protein interaction network, looking for small subnetworks to be topologically significant with respect to all of the targets in our seed. Okay, now this is kind of how our computational model. We basically take all of these seed targets, map them to the interactome, and then we systematically go through each node in the global protein-protein interaction, looking, basically making an assignment for each node. And we assign its status in one of 3 ways. Every protein in the interaction either becomes an interactor, that is a protein which directly interacts with one or more of our seed targets, or it becomes a crosstalker. And a crosstalker is a node in the protein interaction network which is significant with respect to crosstalk to all of the seed, okay? Or of course, many nodes which are neither. So when we simulate those small subnetworks, we can basically reach back into the our model is complete, and for each one of our disease targets, we can pull out of the interactome, and out with it will come either a small subnetwork of interactor and or a small subnetwork of crosstalkers. Okay? Once we've assembled these candidate subnetworks, we can then score them in the method similar to what Trey Eidecker did on the previous slide. We're going to score them for differential dysregulation between consumer using a measure of mutual information based on microarray profiles in colorectal cancer tissue. Okay? And these networks are going to be used to drive in silico validation via classification or in vitro validation by a method that I'm going to show you in a moment. So here is the model data we used. For protein interaction data network, we used HPRD. I believe this was a version 6 copy. It encoded 9,317 proteins exhibiting at least one interaction. One of the proteomic data sets we used was one I just described, obtained in our lab, 72 targets, 60 of which mapped to HPRD. Another publicly available proteomic set of disease targets was produced in the Richard Caprioli's lab by Dave, who was then a postdoc in his lab. There they found 55 targets significant for colorectal cancer, 37 of which mapped to HPRD. And a little later, I'll share with you another seed we used to compare this to a genomic-driven approach. Here, these were 59 highly mutated genes found significantly mutated in human colorectal tissues, published in a landmark science paper in 2000. Those of you familiar with this area, these are the so-called CAN genes in human colorectal cancer. 42 of those mapped to HPRD. For scoring our subnetworks, we downloaded 2 microarray data sets from the Gene Expression Omnibus. One of those was done on an Affy chip, 32 tumor samples with matched controls. Another was done on Illumina, 48 experiments, 24 tumor biopsies with matched controls. Okay, and here's what the data look like. This is one of the selected results. This is using the seed obtained in our lab of the proteomic targets, scored with the Affy array. Now let me orient you to the graph a little bit. Here is our measure of coordinate dysregulation, mutual information, as I mentioned to you before. Here is the subnet size on the x-axis. The blue line represents the mean coordinate dysregulation of a set of subnetworks of equivalent size and of equivalent topology. Okay? The vertical lines represent one standard deviation away from the mean. So we plotted only those significant subnetwork crosstalkers which were significant. That is everything above the standard deviation line here. Those are denoted in red. And then we also plotted these significant interactor networks, which are shown by the green squares. The labels indicate the name of the proteomic seed that actually condensed that network by our network cross model. Okay? Basically, what you can see from this is that most of the seeds did not condense a significant subnetwork. But a number of them were quite high in terms of coordinate dysregulation. And in a few slides, I'm going to talk a little bit about this one. This is an example using the seed produced in the Friedman lab. Again, we see a similar number of crosstalkers showing up as significant by the measure of population. In nearly all cases, the crosstalkers were more significant than the companion interactor subnetwork, and this was something we expected, but it was not always a guarantee, depending on what microarray we used to score. So this is some indication that we are finding things that are significant. Here is a summary slide. This is the Nibbe seed I showed you, sorted by the 2 microarrays. This is the data for Friedman. In general, this is just a summary of what I've already told you. Most of our disease targets did not nucleate a significant subnetwork by measure. And there were a similar number of interactors and a similar number of crosstalkers across data sets, indicating some robustness of the model across different seed types and across the network cross model in the implemented in HPRD. So I apologize, this slide didn't render as well as I have wanted it to. What we did is we took all of the genes and all of the subnetworks that were significant back in the actome just to play out and see with respect to topology. And again, it's difficult to see. You might take my word for it. The paper illustrates this better. But the orange nodes are proteomic disease targets. The black nodes are crosstalkers, and white nodes are actors. What this shows is what our hypothesis suggested, is that proteomic seeds are not distributed randomly throughout the global interaction network, but they are, in fact, tightly clustered. And this has been shown by a number of papers, that disease genes in cancer, diabetes, and a number of human diseases are not stochastically distributed throughout the interactome, but actually to cluster. So this is something we expected to see. I told you, I hope I did, that the type of approach is to drive. This is because wet bench validation projects are time-consuming and it is very common that in silico methods, a lot of significant results, and they can't possibly all be validated. So we need to rank order. That's what we did. I showed you a couple slides ago the TCP1 subnetwork, which was TCP1 is the T-complex protein. It is a hetero protein with 2 stacked subunits with 8 subunits apiece. Some of those subunits are shown here as the CCT proteins, 4, 5, and they all showed up as crosstalkers in this network, and there are a number of other proteins. So we became interested in this network. The reason it showed up significant by this analysis, but in earlier work we had done in a non-crosstalk network model, form work which we published in Molecular and Cellular Proteomics in 2009. Several of the subunits of this complex also came up as significant, as did a protein called PLK1, which is a polo-like kinase required for proper mitotic spindle assembly in most human cells. This is a protein which is known to be overexpressed in colorectal tumors. It is known to be overactive in colorectal tumors. It is to be a prognostic biomarker in colorectal tumors. And so we became interested in this protein as well. So in a separate cohort, independent samples not used in the proteomic screen, we wanted to validate the activity of this small network that I showed you on the previous slide. And so that's what we did by Western blot. A separate cohort of samples labeled as normal tumor, normal tumor, normal tumor here. This is the Western blot data for select targets that appeared in that network. And as expected, most of the targets are over 2-fold. Some as great as 9-fold or even greater than 10-fold in some of the patients. And this is what our model predicted, and this is what we expected. So we had thought this would be overactive with respect to the tumor, and this is in fact what we found. PLK1 is up over 3 to 6-fold, something we didn't expect. After publishing this study, we started doing a bigger literature search on this. And PLK1 is now an inhibitor in phase trials for most solid tumors on Cova Therapeutics. At the level of the message, what we did is took the mean fold change for the microarray data at the level of the message and plotted those. And as I think you can see, there's a very modest increase, if at all, in any of these proteins at the level of the message, suggesting that by just a genetic approach or just an expression array approach, these changes likely have gone unnoticed. Now, earlier I told you, actually, the reviewers wanted us to compare this to a genetic-driven approach, so we went and said, okay, we'll take the genes most highly mutated in colorectal cancer and use those as a seed for our approach. And this is what we found. You already have seen these graphs before. We did this for both arrays, scored the subnetworks with both arrays. That's what's shown here. And there's a couple interesting things about this slide. Number one, the number of crosstalkers or networks at all, there were no interactors which showed up as significant, number one, when scored on the Affy array. There were only 4 crosstalkers which showed up as significant. They were generally not as high in terms of their coordinate dysregulation as the proteomic subnetworks. As sort of a positive control, the top scoring subnetwork was APC. APC is widely held to be the gatekeeper gene in the pathogenesis of colorectal cancer, so we were happy to see that one. Strikingly, every single crosstalker subnetwork that appeared by scoring by the Affy array appeared by scoring on the Illumina array, suggesting again the robustness of our scoring method across different microarrays. In a cross-validation approach, we use the subnetwork as features to train an SVM-based classifier to classify tumor versus control. We did this in a cross-validation approach. That is, we trained on one microarray and we tested on another. And here are the results. So for the proteomic crosstalkers, I think you can see that, well, the first thing to notice about this slide is that the cancer driver genes, their crosstalkers, performed quite well in classification. This is the number of subnetwork features. This is the F measure, precision times recall. The CRC driver genes actually performed quite well, those subnetworks that were nucleated by the genomic approach, as we expect they should, because the seed enjoys genome-wide coverage. Whereas proteomic profiling, we cannot profile the entire proteome for all proteins up and down in the cancer. So that was not unexpected. However, what was somewhat unexpected is that the proteomic crosstalkers, regardless of which array you look at, actually performed better for 3 or fewer subfeatures. 3 or fewer features. Now, this is important because with small cohorts of data like we're working with here, it is important that we have a small number of features, otherwise we run the risk of overtraining the network. So this is actually a desirable feature. The purple line is what we found when we took a traditional approach. We rank ordered the genes on the microarray and used them to train the classifier, and you can see their performance is significantly less better than any of the network-based approaches. Okay? So in summary, how am I doing on time? In summary, I think what I show is proteomic targets significant for a complex disease phenotype are useful for scoring subnetworks for discriminating disease phenotype. The candidate networks implicated in a disease, these provide a practical basis for mechanistic follow-up for validation and for the role in disease. We found that the proteomic crosstalk subnetworks were strong classifiers of the tumor phenotype, and an in silico validation actually superior to the genetic approach. And the overall systems biology approach provides for a nice framework. We are cutting up these methods, and we'll have them available for a wider use on our lab website probably within 6 months for those of you who are interested. By saying that we are colleagues of my lab are hosting the next PacBio conference. This will be held in January. You can all imagine yourselves sitting on the beach in Integrative Proteomics for translational science, and we have a call for papers interested in this area. Please consider submitting a paper. I noticed that passed, but if you mail Gherkin in the lab, I'm sure he will still consider. Okay. So with that, I will make my acknowledgements. I want to thank Mark Chance, my mentor, Salim Chowdhury for help with the classification experiments, Maya Turk for many, many fruitful conversations, the Case Comprehensive Cancer Center for providing us the disease tissues. The funding comes from these agencies, and I would really like to thank the ISB for the travel fellowship and for the opportunity to present at a time, and I'm not going to be able to present future work. With that, I will say the moral is, when you're looking for disease culprits, make sure you're in the right neighborhood. So I thank you for your attention.
Got time for questions? One, you showed the overlap of all the experiments, and the overlap was zero in all these experimental
Correct.
experiments. I see very often now when you compare experiments. Can you comment on that? Is that okay, or do we measure?
So the question is, when I showed the Venn diagram, our model data, there was no overlap of the targets amongst the mutated genes and any of the disease targets that we found by proteome profiling. Now, this is something that's not unexpected because, well, number one, by proteomic profiling, we don't enjoy whole proteome coverage. That is, we can't scan the whole proteome to look for prognostic targets. But the second is that in cancer, it is frequently the case that genes which are thought to be drivers of cancer aren't overexpressed, and so they do not show up as differentially expressed either at the level of the message and sometimes frequently at the level of protein. The reason they're thought to be driver genes at the level of the proteome is because they're differentially active, not so much they're differentially expressed. So that was actually not a surprise.
Okay. Thank you again.
Thank you.
There are no more questions.